Okay, very good. So here we go. I think I will, it's a little shorter time than I expected, so I will go over things quickly. First, this is sort of content. I'll talk about plasma proteomics, antibody reactivity, large-scale blood sampling, and spatial functional proteomics, and I hope I can convey that. And I also have to tell you that I'm conflicted in that I'm affiliated with companies. Olink I'm no longer affiliated with. They are acquired by Thermo Fisher, but the other, Sampifax and Da Vinci, are ongoing company interactions for me. So I wanted to start off with this very old pictures. Many of you are too young to have seen these, I think. This is the old Octoloni technology, which I think is a beautiful technology. It gives you a very vivid picture of what antibodies are like, what they do. You put your antiserum in the middle hole, and then you put various antigens in the other holes, and then you allow them to diffuse till they reach a point where they form an aggregate and precipitate, and that's what you're seeing. The lines are aggregates. And you can see all sorts of interesting phenomena, little spurs. If you look at the lower right, I don't think I have a laser pointer myself, but at the lower right you see this little spur, which indicates that maybe the antibody can bind two antigens. There's a cross-reactivity element, and cross-reactivity is something I will talk about, or how to deal with cross-reactivity, and I guess that's a topic for the whole conference. So antibodies do exhibit cross-reactivity. All antibodies bind more than one thing, for sure. They can still do their task. It depends on how abundant your protein you're looking for is. If it's a very abundant protein, then you can tolerate a lot of cross-reactivity. If it's not, then you may be in trouble. But the very simple solution to this is to ask two questions of your protein. So the first antibody does have some cross-reactivity. It finds its red target, but it takes a few other molecules, and then you add another antibody. That's a very simple idea. Again, going back to history, I should first say that another aspect is how well antibodies bind, which is something, this is sort of an idealized curve of you add your antibody, it binds everything that it can bind, and that's not reality. So you have on rates and off rates that you have to pay attention to, but that's sort of beyond the scope of what I'll talk about. But I wanted to bring up this old paper from 1960, the Nobel Prize earning paper from Yalov and Burson, where they demonstrated the radioimmunoassay and the possibility of detecting protein with a competition assay, a single antibody. In my mind, that wasn't such a revolution, but I think what came later from my home city, Uppsala, was the sandwich assay shown below. So you have a capture antibody, you add your protein, and then you have a detection antibody. And now you get much better specificity, and that kind of dominates, has been dominating how we measure protein, although there is more competition lately. So this is a cartoon. Maybe I don't need to go over it, but to the left you have the single binder assays, forward and reverse immunoassay. You have the sandwich immunoassay on the C. D is Quanteric's Simoa assay, which is a regular sandwich assay, except that the single antibody is detectable. So and only the antibody can give you enough signal. So you have a single molecule detection. That's why it's single molecule assay, Simoa. Then you have the SomaScan assay from SomaLogic, which is very unusual and different from others. It uses aptamers, as you know, and it requires first that they capture the protein, and then you biotinylate the protein, and then you release, and now you capture the biotinylated protein, and you detach the attached aptamer and read off the identity of it. And that turns out to be highly scalable and sensitive. But then on the two right, you see the proximity assay, the proximity extension assay that's made by Olink Proteomics. And I just illustrate one version where you have a capture antibody first, and then you do the proximity ligation step. Just one word, I think others will maybe talk more about Olink, but and I believe most of you are very familiar with the Olink assay, but it's two antibodies with conjugated oligonucleotides or antibody preparations. Often it's the same polyclonal antibody that's split in two. Sometimes it's monoclonal antibodies. And they can recognize the target. And if they do recognize the target, then you just dilute the sample. You don't wash anything. You just dilute it so that the chance that two antibodies will be in proximity is lower. And then you allow the ones that remain in proximity to extend and give you an amplifiable molecule. And I think currently they are measuring 5,400 proteins in 2 microliter of plasma and doing very well. And I just put this slide in with some data. 2 years ago, in August 2023, there were 3 back-to-back papers in Nature describing the first 50,000 individuals from UK Biobank analyzed with respect to 3,000 proteins. It's interesting to see that how that has given rise to a whole literature of people just sorting through that data, not doing any new assays, without any new patient samples, just using the published data and finding a lot of very interesting things. So in the middle panel, you see 200 of those 3,000 proteins turned out to correlate strongly with age. So you could tell the age of a person, but you could also tell the little yellow spot above the diagonal. I really should have a pointer laser I could use. No. No. So. I can use that. Okay, good. Yeah. So you see these little points up above the diagonal. The people who have the 10% oldest protein compared to their chronological age, they can have a 5 times higher risk of dying in the course of this study, or 4-fold higher risk of cardiovascular disease, diabetes, and so forth. So you really need to keep your proteins young. On the right, you see another paper from early this year, just demonstrating what kinds of conditions, infectious disease, neoplasm, and so forth, different proteins correlate with. And you see up top here some highly significant associations between protein. So we're building a large body of knowledge about what proteins mean right now. And I think that's very exciting to follow. I'll just say a few words about version. We have inverted the PEA assay. So at the top you see the regular PEA assay. Now we have also conjugated oligos to antigen, and now we're looking for antibodies that are able to bring 2 copies of the antigen in proximity. And if so, then we can detect that by the same means as in the regular PEA assay. So what we're after is antibody specificities. Of all the things that you can measure in blood, what antibody specificities you carry around is one of the more significant ones. It tells you if you have an autoimmunity, it tells you if you're properly vaccinated, what infections you have suffered. So using the very same format, we can do this. And I just show these are a couple of 4 well-known autoantigens in systemic lupus erythematosus. And as you can see, even when you're down to 1/1,000,000th of a microliter, many patients still have detectable levels of antibodies. So it's highly sensitive assay, highly multiplexable, and you can measure antibody reactivity. Here's a slide where we looked at 17 different antigens in 16 blood donors and 22 SLE patients. The pinkish is SLE patients, and you can see that they stand out. So we're able to screen for autoimmunity or for what kinds of infections you have suffered very quickly in the same technology. I'll go on to a little different topic. So my lab is mainly making what we call molecular tools, often built on oligonucleotides that get ligated or sometimes polymerized. But we realize that there's so much we learn about protein patterns and antibody patterns, as well as, of course, of transcriptome and so forth. So we really the old genetic paradigm was that you needed one sample from every person and then you have all the information you need. With proteins, you want to have regular samples. You need simpler ways of collecting samples. So we developed another approach. First, in 2017, we demonstrated that if you put a drop of blood on a piece of paper and then you prick this piece of paper and punch out a square millimeter of paper with blood on it, then you can measure an Olink panel. So these are Olink panels. These are duplicates in blue of wet samples, and these are duplicate in red of dried samples. About the same sensitivity, very good reproducibility, horizontal line, not very good correspondence. So some proteins are more detectable in the dried state, some less detectable. But as long as you compare dry to dry, it's an excellent way of measuring proteins. And we developed a little card, which I'm advertising, and now I'm talking for a company. This is a little card, credit card size card. It rotates a little bit too long, but eventually it sits down on the page. You wash your finger, you prick your finger, put a drop of blood on top of it. All the cells stay on the top membrane. the plasma membrane is processed underneath, and the plasma is carried by capillary forces. It takes about 1 second. 60 seconds later, it's dried, and now you can send it in by mail, and you can now have a robot take care of it, and a robot can punch out the square millimeter and deposit it in tube, and do the Olink assay or measuring transcriptome or whatever you want to measure. So it's a possibility of building really large consecutive biobanks, which I think we really need to understand better how these factors. So these different plots show you that it's reproducible. If you have two, on the left here you have, on the left you see a correlation between two samples, pretty good correlation with low CVs in the single digits. If you compare wet plasma with dry plasma, then correlation is not so good anymore, so you shouldn't do that. And also we all get a cell fraction that you can measure protein or transcripts or the genome if you want. And then again, the correlation between wet and dry is not perfect. So you need to compare wet to wet or dry to dry. Finally, I'll talk about spatial and functional proteomics and how we can use these molecular tools to detect protein, image them, but also to understand what proteins are up to. And proteins are, of course, the real agents that do conduct activities in the cell. So we really want to know not just where they are, but what are they up to? How can we measure their activity states? And this is just one image where we are looking for E-cadherin and beta-catenin, and I think these are gorgeous pictures where you can get an overview of mucosa, intestinal mucosa, and then blow it up and see more closely what happens. So spatial biology has a lot to offer. However, it has been a very stagnant field, spatial biology. So this is a paper from 1941 describing the possibility of putting a fluorophore on an antibody, adding the antibody to a tissue section, wash away what didn't bind, and then looking at it. And since 1941, that's pretty much what we're still doing with immunohistochemistry, immunofluorescence. We have had revolutions in how we measure DNA and revolutions in protein measurement in solution, but this still people are doing this and are completely victim to the specificity of individual antibodies. And as this whole meeting, I think, is about, you can't really trust antibodies. They're like children, you can't trust them alone. So here's an example. Here we did the capillary electrophoresis. And we compared a rabbit polyclonal and a mouse monoclonal, and then we detected it with secondary antibodies. And both the rabbit and the mouse find aldose reductase here, but the mouse sees something over here, and the rabbit sees something here. If we use rolling circle to amplify the signal, then we get an even messier picture. However, if we say we're only going to trust this if the rabbit and the mouse agree, then we get this nice pristine peak and nothing. It's a very clear illustration of the value of dual recognition. Same for S100A6, cross-reactivity pattern, even worse when you just amplify it, but very clear. And in the third case, we have two rabbit and a mouse directly against CDC34, but they can't make their mind up. The mouse say, here's the protein, rabbit says, here it is. And if you now ask them to agree, well, you get nothing because one or both of them are wrong. So you really want to use pairs of antibodies. I have a series of examples. This is two monoclonal antibodies against RAF1. Pretty much everything is in the tonsil is brown. Some things are more brown than others, but it's pretty messy. The other monoclonal antibody, well, a little worse maybe. But if you use them both together in immunofluorescence or using the in situ PLA technology, then you get very nice labeling where the label should be and not elsewhere. I think this is really the way one should do it. Here's another example, two monoclonal antibodies against VCAM. One, a lot of background, a little less background, very clear picture when you ask both to agree. A third example, messy monoclonal antibodies, clean when you ask them to agree. the final example. But not only can you enhance specificity, but you can also look at more what are the proteins up to. And some of you have heard me talk about this for many years. So we study things like where the proteins are interacting, and we study if they have been phosphorylated, and if the phosphorylated proteins are interacting. In the top is whether they are phosphorylated, and the bottom how the phosphorylated protein is SMAD2 is interacting with SMAD4, and when it peaks 90 minutes after TGF-beta, and we can count and get digital counts, perfect for AI, having digital counts of cell reactivity. Here's an example from some local colleagues, Karin Strell and Patrick Micke. They used the in situ PLA to study lung cancer patients, comparing expression of PD-L1 as a predictor of responsiveness to treatment with immuno-oncological treatment without being able really to tell the patients apart. However, if you look for interactions between PD-1 and PD-L1, then patients with high levels of interaction are more likely to respond compared to the ones that have low level. So you learn more about proteins, not just where they are, but where they are talking to each other. This has now been expanded by the company Navinci, which I'm affiliated with. So they are looking at PD-1, PD-L1, PD-1 interacting with PD-L2, PD-1 interacting with SHP2, and some other interactions. And you can get all this together. So you add all antibodies together, make the rolling circle products, and then you read them out all at once if you can distinguish that many colors, or a few at a time. Remove the signal and look at the next set. So we did that for EGF signaling. So in the EGF signaling cascade from the surface receptor all the way till transcription is activated in the nucleus, there's a number of relay stations. So we have here 18 antibodies, 9 antibody pairs that look at various reactions. All are added together. You generate DNA circles. If they bind in proximity, you amplify the signal, and then you read them out. We only detect 3 colors, so we read out the first 3, remove the signal, next 3, remove the signal, the last 3. And this is what it can look like. So here we have looked at cell lines that have been breast cancer cell line that was unstimulated and then stimulated. And we look at JAK1 interacting with JAK3, JAK2 with JAK3, JAK1 with STAT3, JAK2 with STAT5A, and so forth. So you learn a lot about what the proteins really are up to. And here's another panel where we are looking at phosphorylation. So again, we don't trust that an antiphosphate antibody also will be specific for the right protein. So we take a phosphor-specific antibody and we combine it with an antibody that we trust to detect the protein. And only when they bind together do we get the signal. So this is the unstimulated, and the stimulated are much more active in phosphorylation. And this is doing this now in patient tissue samples. So these are a bunch of lymphomas, Hodgkin's lymphomas, and a B-cell lymphoma, and a thymoma, looking at various protein interactions. So I really think this is what we should be doing. Transcriptome gives you a lot of information, but looking at what are the proteins up to really is what you want to learn. Final example, we can also replace one of the antibodies. Here are 2 antibodies against the EGF receptor, giving rise to a circle that gives you this red staining in breast cancer tissue. But here we replaced one of the antibodies with a low molecular weight drug, a kinase inhibitor. You just put an oligo on the kinase inhibitor. And if they bind together, the antibody against the EGF receptor and the inhibitor, then we get the same red signal. So this is what is known as target engagement. In pharmacology, you want to know, does my drug hit its target? And here we can see that. Here we use not gefitinib as a drug, but erlotinib, which is a good kinase inhibitor that should bind the EGF receptor. However, the way we put the oligo on it, we destroyed its ability to bind. So it's a negative control, and it's very negative and nice. So with that, I'll just end if I can. Let's see. So I'll end and thank people in the lab. There's been a fantastic group of people behind all this. Simon Fredriksson is really a person very centrally in the proximity extension assay. He was the CEO of Olink Proteomics for a while, or Olink Bioscience before it became Olink Proteomics, and had a very important role. Ola Söderberg was very important in the in situ PLA, but there's been many very talented students behind this. And here's a group of more recent students. And here's a picture of us at with the Baltic Sea, and you see a little bit of the Baltic Sea behind there. Anyway, with that, I'll close and take any questions you might have. Thank you.